Hey guys, good morning. So I was planning like all week, literally. I was like, oh, I'm going to see my students Friday. So, that's kind of what I've been telling you guys all week. And then it literally hit me like 5:45 this morning. Wait a minute, I don't see biology, first period biology on Fridays because I don't get there because I gotta drop Coral off at treatment at 9. And so I try to drop her off a little early and then race over to you guys. So, anyways, this is the plan for today. There's one of two things happening, and I'm not 100% which one is happening. So, you guys will have to, yeah. So, if there is a cart of microscopes, oh my gosh, I forgot to hit play. No, you should be fine. This is recording a different way. I just suddenly saw this and I was like, oh, I'm not recording. No, I should be. Anyways, if there is a cart of microscopes in the classroom, if Mrs. Morehart brought the microscopes today, you guys will be doing, honor students will be doing the lab that you already have a copy of. And I'm going to switch screens here in a moment and walk you through that lab so that you are understanding what you need to do. If there are not any microscopes in the classroom, it means that you can't do that lab. And I will make sure the microscopes make it in there. And then whatever day you happen to have the microscopes, then you'll do that lab. You'll just pause wherever we are in learning of the photosynthesis, and you will do your algae lab for honors. My non-honor students, if the algae lab is there, you can do one of two things. You can just kind of use this time as a study hall to catch up on things if you are missing anything for me. I spent like 4 hours grading last night, and I still have some more grading to do. But there's, interactivities. There's the worksheets you guys have been doing, like the specialized cells and transporting cells, things like that, making sure all that stuff is with me. Or I have the next set of notes already on Classroom. Now, again, I'm not there. I'm going to print them off for second period. I figured I'd have them for you, but I'm not there. So print off the fill in the blank notes, print them to the classroom. When you print them only on one side and print them at 90% and then trim them down. Okay. So honors biology. Let's go through the algae lab. But first, if there are no microscopes in the classroom, you will start the photosynthesis video that is on there. Now, it will take 2 periods to get through that photosynthesis video. It is about an hour and a 15-minute video, so it will take you 2 full class periods to do that. So go ahead and start watching that. I will send the link to Mrs. Angulo so that you guys can watch it as a class. If for whatever reason that hasn't gotten set up yet with the sub, that's totally fine. It's on Google Classroom. You can watch it on your own. But again, print out the notes, single-sided at 90%, and then trim it down. There is 2 videos on there, and I'm trying to get a third one up, but Savas stopped working last night, and I could not get the e-text figure to launch so that I could walk you through that, which I had told you I would do, and I wasn't able to do that. So those are the 2 things. If there's microscopes in the classroom, honors biology, you are going to do the algae lab, which I'm going to walk you through in a moment. If there are no microscopes in the classroom, then you guys are going to start the photosynthesis lesson. And then whenever the microscopes show up, whether it's Monday or Tuesday or whatnot, just you guys don't do whatever I have assigned that day, and work on the algae lab. Okay, does that make sense? So I'm going to switch screens real fast. Okay, so this is what you guys have, the lab investigation here. What structures make up algae cells? So, what are some characteristics of algae that make them useful for producing biofuels? So, introduction. The term algae describes a wide variety of relatively simple organisms that convert sunlight into energy. Algae can be either unicellular or multicellular, and they are found in freshwater and saltwater. Like plants, algae contain chloroplasts that allow them to convert energy from the sun into storable chemical energy. When conditions are right, algae can store energy in the form of natural oils. Scientists are researching different varieties of algae and developing practices to grow algae efficiently. In the future, algae could be a very common source of efficient fuels. In this laboratory activity, you will examine algae cells underneath the microscope and make drawings of what you see. You may use prepared slides or obtain your own algae samples from a nearby pond. It's winter, so we're not going to a nearby pond and don't really have a prepared algae set. So what you guys are going to do is I have a really great fish tank in my classroom that has algae in it. So this is what I want you guys to do. So you have a microscope and it says per group. So that's kind of up to you. Everybody will have to turn one in, but how many people want to share a microscope? It depends on how many microscopes we have right now. Also, if there are not any microscopes today and you can't do this lab today, feel free to rewatch this portion of the video when the microscopes actually get there so that you can have a refresher of what you need to do versus me making a new video. You can just rewatch this video. Okay. So this is what you're going to do. You're going to need your microscopes, your packet. You are going to need a dropper pipette. Those are in a drawer. There's a pipette drawer, big surprise, right? So you're going to get a pipette drawer. You're going to get a glass microscope slide and a cover slip for a microscope. And a cover slip is like this big, and it's plastic. Okay, so where are these things? Things get moved around in my classroom throughout the week while I am not there. But there is a box and it's about 18 inches long and it's only about 6 inches wide. And it's just a cardboard box. And I had it on the back table. So I'm hoping that it is still there. But if you look inside of it, you will see some prepared slides. There just aren't any algae prepared slides. It's the slides you guys looked at before. But then there are going to be blank, like empty glass slides. And then you will see boxes for cover slips. So you guys will have to find that box. It is somewhere on a countertop, on the floor, in my classroom. It should not be in any cupboard because I don't have any space in any cupboards. So it could be on top of the big metal, my big metal double door cabinet. It could be on the back table. It could be on the floor under the windows. It could be over by all the equipment. I am not 100% sure because things get moved. I leave on Sunday. I come back on Friday. Things are in different spots. Find that box. That is where your slides and your cover slips are going to be. I would prefer for everybody to have their own. We have plenty of slides and cover slips. So what you're going to do is you're going to take your microscope slide and your cover slip. And I would like one person, preferably one, to get a pair of gloves on. And the gloves are in the drawer right behind Kelsey. Okay, you'll open it and there should be gloves in there. Fingers crossed. If there are no gloves left, just run down to the office and borrow a pair of gloves. And I want that one person to gently, like, make sure the fish tank light is on, but gently open the glass lid and then actually pull out some algae, not the plants. So you can do a couple things. There should be some algae like growing. If there's green on the glass, then that's algae and you can somehow scrape that off. You can actually probably use the cover slip because it's plastic, and get some, or if you find some like longer algae, just like get some gently off of the leaves of some of the plants in there so that you can actually see green stuff. You can also just pipette some of the water, and we can have people do a couple different things because all of those algae, the stuff on the plant, the stuff on the glass, and the stuff in the water are going to be different types of algae. So it's fine doing any of this. I just want to make sure you can actually see the algae structures. Okay. So what you do is you're going to put, so you're going to get the algae out of the tank. You're going to put it in the middle of, so your glass slide is about like this big, and you're going to put it right in the middle. And then you're going to put the cover slip on top of the algae. And that's what holds it in. It's fine if there's water and stuff, just put it on top of the slide like that. And you can kind of, with a paper towel or tissue, wipe off any of the extra water that is outside of the cover slip. And then that's your slide. Does that make sense? That's pretty simple that way. There is algae in the fish tank. Yeah, there is algae in the fish tank. So, and there's even some on the wood and various places. Okay. If somebody has a really good slide, and once they're done, you guys can share and see different things. That's fine. Okay, so let's go back to the slide. Here we go. Okay, so that's what you're going to do. Okay. You don't really need safety goggles. If you're not touching the slides, you won't need gloves. But if there's enough gloves, feel free for you guys all to wear a glove, or at least wear a glove on the hand, that your dominant hand that you're going to be handling everything with. And then do make sure that you wash your hands really well. Okay. If you have to move tables around to get microscopes plugged in, that's fine. But please, 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 please put them back, because second period biology is not doing this experiment. Make sure your microscopes get back on the cart, so that Mrs. Morehart can take them back. Okay. So you're making your slides. There you go. It's kind of what I told you. And then simply start at the part B, making your observations. Okay, carefully lift the slide and place it on the stage of the microscope. Start with the low power, slowly move up as you can, and then move up to the highest one that you can actually still see stuff. Okay, and then draw what you see there. You guys have done this before. Label any structures you can identify. So can you see a cell wall? Can you see a nucleus? Can you see chloroplasts? Can you see any structures that you learned about? Okay. And then once you're done, you're going to completely clean and dry the slides and cover slips. They're reusable. Just put them back where you found them. Just go to the bathroom, go to the kitchen, be gentle, because you can break both the glass slides and the cover slips. The cover slips are plastic. I'm not as worried if you break those. I'd like you to not. But the glass, if you break the glass slides, you can't throw them away. Do you understand that? There is a bucket in the bottom drawer, I think it's the bottom drawer, of my black filing cabinet. There is a bucket in there that is for broken glass. You must put the broken glass slides in there. Do not, under any circumstances, throw them in the trash can. That is not laboratory safety. Okay? Makes sense. And then you're going to kind of answer all of these questions. They extend your inquiry. Algae can be used as biofuels. What properties of algae do you think make them useful as biofuels? What properties might limit their usefulness? Support your answer with observations from this investigation and logical reasoning. You can absolutely do that question. So go ahead and do that. So, like I said, you're doing that lab if there are microscopes. Obviously, I'm hoping even after this long video that you are able to get that lab done in class, at least the observations of the algae cells. You can always answer the questions once you're done looking at the algae cells and once you have everything sketched. So really just be efficient in sharing the microscopes and making sure that you get your stuff drawn so that you can move on to the question so that everybody can preferably get the microscope stuff done. It makes me nervous to leave the microscopes out all weekend because there's people are in and out of my classroom and I just don't want anything broken. Microscopes are a very big temptation to want to handle and touch and play with. Not that anyone I think is going to intentionally damage anything. I don't believe that by any means. But accidents can happen, and I would prefer to just not have to have them out. And I just don't want to have Mrs. Morehart have to get them in and out of storage constantly because they are in storage. They are not super accessible. But if there are no microscopes, you are to do the, let me go ahead and get that open real fast. Look at all this stuff I have to. You're going to go ahead and there is notes right here. So there's the video you're going to watch. I mean, there's just notes. There's the video you're going to watch. And there is the second video. The other thing that you're going to do is what I told you right here. The test question explained. You guys watch that video. I have already changed the status so that you can go on today and adjust that question as needed. If you don't have time to do it in class right now, that's perfectly fine. I get that. So go ahead and just do that at your own time. But by the time I grade it, it needs to be done. So does that make sense? Okay. So thanks, guys. And